Okay, this week in lecture was kind of a hodgepodge. They went through basically two things that kind of fit together, but I think it could have been organized a little bit differently, but we're going to go ahead and go through it anyway. Anyhow, the first thing is control of pyruvate, so pyruvate dehydrogenase. And so that was, there are two things. That was the first thing. The second thing was the fate of oxaloacetate. Oxaloacetate. So the first thing you got to think of is energy charge. So these are key points. Energy charge of the cell. And by that, I mean if the cell has a lot of ATP or a lot of reducing equivalents, NADH, then it has a high energy charge. So if those are high, its energy charge is high. If these are low, then the energy charge is low if it has ADP or NADP. ADP, NADP plus, then the charge is low. The second thing is the state of the, so this is the energy charge of an individual cell. Then we got to think whether it's during the well-fed state versus the starvation state. So well-fed versus starvation. And sometimes starvation seems extreme. I like the word fasting. And this is more of a systemic thing, systemic. So why is that important? Well, if you are in a fasting or starvation state, you can still have a high energy charge in specific cells. For example, in the liver, if you're in a fasting state, you're breaking down, you're mobilizing fatty acids from the adipose tissue, and you're sending them to the liver. And then the liver is oxidizing those, creating energy out of them. So the energy cells have a high energy charge even in a starvation mode until you run out of fat or amino acids to keep the energy up. However, that happens so that you can divert specific compounds into gluconeogenesis. So let's take a look really quick at pyruvate. So we got pyruvate, and it can undergo, so it can be acted on by pyruvate. So pyruvate dehydrogenase. I'm just going to put dehyde, and this is abbreviated PDH. So pyruvate dehydrogenase and PDH are synonymous, and that turns it into acetyl-CoA. And the other thing that can happen is it can be acted on by pyruvate carboxylase, so PC, pyruvate carboxylase. And actually, that should have been drawn right here. So this would be PC. So this is the enzyme, and that turns it into oxaloacetate. So if we just trace this out a little bit, so if it goes to acetyl-CoA, it can either be used for lipid synthesis. So we can say that this is creating lipids synthesis. Fatty acid synthesis is another way of putting it. Or it can be converted to CO2 and energy. And then the lipid synthesis can also be run in the reverse direction through a different pathway. Then there's another reaction that pyruvate can be involved in. So let's say that pyruvate is converted to alanine. So this is where glutamate kind of interacts with this and forms alpha ketoglutarate, and then this can actually become oxaloacetate as well. So kind of big picture, what we're looking at is what can happen to pyruvate. So it can go through a pathway to become acetyl-CoA, it can go straight into oxaloacetate, or it can be converted to alanine as glutamate goes through the pathway to oxaloacetate. And we're going to talk about some of the controls of this pathway of pyruvates, of the fate of pyruvate here in a minute. First, we're going to talk about oxaloacetate. Just as a short summary, oxaloacetate can be sent to the TCA cycle whenever there's a low energy charge, or it can be sent to gluconeogenesis. I'm just going to call it GNG. That's not really a technical term, but that's under the high energy charge, or it can be used for to make citrate, and then citrate gets used in fatty acid synthesis. And this is also under a high energy charge. So we've looked at what happens with oxaloacetate, its TCA, gluconeogenesis, or fatty acid synthesis, and how that relates to the energy charge. Now we got to look at how these things also affect pyruvate. So in short, is it going to become oxaloacetate, or is it going to become acetyl-CoA? What would cause pyruvate to want to become acetyl-CoA and further on to CO2 and ATP? Well, if we don't have a lot of ATP, then you want to make more. So pyruvate dehydrogenase is activated by low ATP and high ADP. It's also activated by high levels of pyruvate. Then it's deactivated by high levels of acetyl-CoA and high levels of ATP. Now we got over here that lipids can become acetyl-CoA, so that's key. This is called beta oxidation. Beta oxidation is when a lipid becomes acetyl-CoA, and what that essentially does is it will inhibit pyruvate dehydrogenase. The reason it does that is because beta oxidation provides energy for the cell. So if you're in a starvation state, like we were talking about, starvation state, fatty acids will undergo beta oxidation and become acetyl-CoA. In the process, they produce a lot of NADH, and through the acetyl-CoA, they enter the TCA cycle and produce a lot of ATP. So that will inhibit the pyruvate dehydrogenase. So we know that pyruvate can become acetyl-CoA, and that acetyl-CoA can either become a fatty acid or CO2 and ATP. Now, this happens because pyruvate dehydrogenase, it can either be phosphorylated or it can be dephosphorylated. Phosphorylated makes it inactive, and dephosphorylated makes it active. So under insulin signaling, whenever you have insulin signaling, insulin indicates, hey, we already have a lot of blood sugar, so don't turn into glucose. Don't do that. So what's its other option? Well, it makes sure that pyruvate dehydrogenase is turned on. So what are we going to do? Are we going to make energy, or are we going to make fats? And so so substrate feedback, the high ATP would feed back and say, don't turn on, but the insulin would go ahead and covalently modify it and help activate it. That would lead to fatty acid synthesis. This is kind of the idea behind the Atkins diet, if you want to put it all into perspective. So Atkins says low carbs, and you can eat as much fat as you want, you can eat as much protein as you want, just low carbs. And so what does low carbs means? Low insulin signaling, and so you're always breaking down fat, fatty acid catabolism, for energy in the liver, and you're not really doing fatty acid synthesis because the pyruvate dehydrogenase is phosphorylated. Now, I'm not actually advocating or saying anything for or against a low-carb diet, paleo, Atkins, whatever have you. All I'm saying is this is the basic theory behind it. And so this kind of summarizes everything I've just said. So you have a high energy state. It's going to activate pyruvate dehydrogenase kinase, which will turn this off. And then these low energy states will feed back to decrease pyruvate dehydrogenase kinase. Then pyruvate dehydrogenase phosphatase is increased, so you can have epinephrine induced calcium release from the muscles, which will dephosphorylate this. Just regular calcium from muscle release, or insulin will dephosphorylate that, which helps with fatty acid. And so the way to think of it is high energy charge or low energy charge. What are the molecules involved and what will they do? And I think this is the high yield way of thinking about things. Is it high or is it low energy? What does that mean? And what molecules indicate high and low energy?